Good morning to all of you. Today's our lecture is about protozoan protists. As you know, we have studied about protozoan protists, and on the basis of locomotory structures, we have studied zooflagellata and ciliata. And yesterday we were talking about sarcodina, and we came to know about AmoebaProteus and left with some examples in Sarcodina. So let's complete some of the examples in Sarcodina. So we have studied about this AmoebaProteus, irregular shaped structure, and does have a prominent nucleus as well as circular contractile vacuole for osmoregulation, and performs pseudopodial movement, or you can say false feet. So we have studied about this AmoebaProteus. The next example we have Pelomyxa, which is also known as giant amoeba. This Pelomyxa is multinucleate, and as you know, AmoebaProteus is uninucleate. But Pelomyxa, which is known as giant amoeba, is multinucleate, and it has 100 to 1000 nuclei in a single cell. It shows sexual reproduction. This Pelomyxa shows sexual reproduction. You have read in case of AmoebaProteus, only asexual reproduction takes place in case of AmoebaProteus, but in case of Pelomyxa, sexual reproduction by syngamy, just a fusion of two organisms. Okay, so sexual reproduction by syngamy in case of Pelomyxa, but only asexual in case of AmoebaProteus. Okay, and the next third one is Olocantha. And Olocantha is a very important point and has been asked many times in competitive examinations. Olocantha has maximum number of chromosomes in a cell, in a diploid cell you can say. So 1600 chromosomes are present in a cell of Olocantha. Most number of chromosomes in an animal cell, because actually we do consider protozoans as smallest animal-like organisms, or that's why we consider them as an animal cell. So they have maximum number of chromosomes, 1600. Then we have Entamoeba histolytica, and this Entamoeba histolytica actually discovered by Lambele, name of scientist. It is monogenetic. You have read about digenetic. Digenetic means two hosts. An organism completes its life cycle in two hosts. But when we do write monogenetic, that means only one host is needed to complete life cycle. So this Entamoeba histolytica is monogenetic. Only one host is needed to complete life cycle, and that one host is human being, intestines of human being. It is dimorphic in nature. Dimorphic means di means two. Dimorphic means it has two different life forms. One is minuta and another one is magma. This minuta you can call it as younger stage, or this one is an adult stage. This magma stage, very important, this magma stage of Entamoeba histolytica is pathogenic in nature. This magma stage is pathogenic in nature and is also known as trophozoite stage or pathogenic stage. So let's come to the structure, just simple as we have seen in case of AmoebaProteus, but only difference between AmoebaProteus and Entamoeba histolytica that in Entamoeba histolytica there is no contractile vacuole. There is no contractile vacuole in case of Entamoeba histolytica. And as you know, contractile vacuole function in osmoregulation. Okay, means to check the concentration level of fluids, ions, okay, electrolytes inside the organism, body. So this Entamoeba histolytica does not have contractile vacuole. Why? Because it is monogenetic. It lives inside the intestine of human beings, and inside the intestine conditions normally remain same all the time. So that's why it does not change. It does not need to change its nature because conditions are always same. So that's why it does not need to have any contractile vacuole just to perform osmoregulation for it. Okay, so that's why, because it lives inside the intestine where conditions are always same, so it does not need to regulate its body according to any condition or any changing condition, like in natural conditions where conditions are changing time to time. Okay, so this Entamoeba histolytica enters inside the digestive system of human being, that means in intestine, in the form of minuta form. This minuta form changes into magma form. This magma form or trophozoite form or pathogenic form actually causes histolysis. Histolysis. Histo means tissue, lysis means breakdown. So it breaks down the lining of intestine. Sometimes you observe blood in your stool. So that may due to the presence of Entamoeba histolytica inside your intestines. So this magma form, which is pathogenic form, is histolytic in its nature. Histo means tissue, lytic means uh, means lysis or breakdown of intestinal linings. So this magma form causes a disease which is known as amoeboisis disease. Okay, so it's all about Sarcodina. We have studied about AmoebaProteus, Phyllomixa, Olocantha, and Entamoeba histolytica. So let's move to the next one. It's Sporozoa. And as we have read about this, Sporozoa does not have any locomotory structure. Okay, like in case of Zooflagellata, ciliata, pseudopodal movement in case of Sarcodina. So it does not show any movement. It's Sporozoa. So the best example of Sporozoa is Plasmodium, which is known as famously known as malarial parasite. It's very famous parasite, Plasmodium, which causes maximum death all around the world. Okay, so example is Plasmodium, which is malarial parasite. It is without any locomotory structures. I have told you, it is discovered by Leveron in the RBCs of human. It has many different species, like we have Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, and Plasmodium falciparum. Among all of these, this Plasmodium malariae is very prominent in South Asian countries, including India. And but this Plasmodium falciparum is most lethal, or you can say most dangerous among all of these Plasmodium species. And this Plasmodium falciparum causes around 44 to 45,000 deaths every year all around the world, especially in African continent. So this Sporozoa, so this Plasmodium actually digenetic in nature. Now here you can see monogenetic, only one host, and here you can see digenetic. Digenetic means two hosts to complete life cycle. So there is primary host and secondary host. Primary host is female Anopheles mosquito. I think all of you are aware of it. And secondary host is human. So how Plasmodium completes its life cycle in different phases. Okay, so this plasmodium completes its life cycle in three different phases. In the first phase, which is known as schizogony, 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 under this stage, this plasmodium divides by multiple fission or by asexual reproduction. So multiple fission occurs during the first phase of the life cycle of plasmodium, which is known as schizogony, and this occurs inside the human beings. So inside the human being, it occurs in firstly when plasmodium enters inside the human being, it firstly attacks liver cell, then the RBCs. मतलब पहले वो लिवर को इन्फेक्ट करेंगे, उसके बाद वो RBCs को. So firstly okay, so firstly they do infect the liver cells, and inside the liver cell, what do we call it is a hepatic. Okay, hepatic schizogony, and when they do infect the RBCs after infecting liver cells, when they do infect RBCs, they are known as erythrocytic schizogony. So two types of infections occur inside the human being in the liver cells, known as hepatic schizogony, and in RBCs erythrocytic schizogony. So first phase in human beings, okay, where multiple fission takes place, and after this second phase, which is known as gamogony. Gamo means gametes. Gamogony means gamete formation, and it begins inside the human being and it completes in mosquitoes. Means fusion, fertilization or fusion takes place inside the human being, and gamete formation, and that gamete gets matured inside the intestines of mosquito, and after getting matured, that matured plasmodium becomes sporozoid. Sporozoid is an infective stage of plasmodium. Becomes sporozoids and reaches up to the salivary glands of female Anopheles mosquito. Then that female Anopheles mosquito actually bites a healthy person, and when it bites a healthy person, it injects plasmodium into the body of a healthy person through its salivary glands. Okay, the third stage is sporogony, means formation of spores or sporozoid, which takes place inside the mosquito. So it completes its life cycle in three phases: schizogony, which occurs in human being, in liver cells and in RBCs. Second one is gamogony, one half in human being, and second half gets completed in mosquito. And third one is sporogony, which occurs in mosquito, where the sporozoid form starts to form. This sporozoid form, which is an infective form, which reaches up to the salivary glands of female Anopheles, and from the salivary glands, when that mosquito bites a human being, this plasmodium along with the saliva of female Anopheles enters inside the healthy human being, and then again first phase, second phase, third phase, so on. Okay, so it's all about sporozoa and plasmodium. We will discuss in detail about plasmodium in the +2 in the next class. And the last one we have slime molds. As the name suggests, mold means they are decomposers. They do drive their energy on dead and decaying organic matter. They are uninucleate, only one nucleus is there, and they have no cell wall, which is the animal-like feature. No doubt they are uninucleate, but when two or more than two the cells of slime molds, when they get fused, they do form a multinucleate type structure. Here you can see these are individual the cells. Actually they are unicellular, but these unicellular cells, they get fused with one another to form a multinuclear type structure, and they do form a mucilage sheath around them in order to protect them. Okay, so this is the mucilage sheath actually uh, forms a protoplasmic type of mass which is known as plasmodium. Okay, when it comes to reproduction, it reproduces vegetatively, asexually, as well as sexually. Okay, so in vegetative reproduction, it reproduces via fragmentation. Means it divides into many fragments, and especially with the formation of sclerotia formation. Sclerotia formation means this slime mold becomes circular and protected by hard cyst known as sclerotia. Asexual reproduction via spore formation, and when spore is formed, that spore actually modifies into two different structures. That spore either forms this irregular amoeba-like structure which is known as myxoamoeba, or forms a spindle-like structure which is known as swarm, SWARM, swarm cell. Okay, so what did I say? Asexual reproduction via spore formation, and that spore is formed, that spore modifies into two different forms, either in amoeba-like form which is irregular in structure and is uninucleate known as myxoamoeba, or a spindle-like form known as swarm cell. This spindle-like form has two flagella, one small in size and one longer in size, and known as whiplash flagella, because it is broader at the end of, okay, it is broader from here and is narrower towards the body. So whiplash flagella is present and known as swarm cell. Okay, so when it comes to sexual reproduction, this swarm cell sometimes loses its flagella and it becomes myxoamoeba after some time. This myxoamoeba again gives rise to the two daughter bodies which are quite similar to that of parent body. And one more way, this swarm cell, say for example this is swarm cell, this flagella is nucleus. There are two swarm cells, they get fused with each other. Flagella, this is posterior end, this is anterior end. So they get fused at posterior end. Two swarm cells get fused with each other at the posterior end and do form a single cell, which after some time changes into again myxoamoeba. Then again same thing happens, two daughter cells, two similar daughter cells. Okay, so it's all about protozoans, protists, zooflagellata, ciliata like in paramecium, sarcodina, amoeba or entamoeba, sporozoa, plasmodium, and slime molds. So tomorrow, the next lecture, we will discuss about kingdom fungi. Thank you.